you. It's a distinct pleasure to be here, and I'd like to thank the organizers for inviting me to make this trip. I'd never been to the Mideast before, and I'm having an absolutely wonderful time. I've met some of the friendliest people I've ever encountered anywhere. And so today I'm going to talk to you about something that I think is a relatively new, although very exciting idea in cancer therapy, which is immunotherapy. And although I say that it's new, it's actually not really new, but it's become to what we would call a tipping point right now, at which the clinical trials are starting to happen very frequently, and it's going to be appearing more and more in the clinics. So just a brief discussion of cancer treatment. The oldest treatments were some sort of surgery. In this timeline, I put a pioneer of immunotherapy, William Coley, that you may have heard of. If you're interested in immunotherapy, you should look him up. The development of radiation in the early part of the 1900s, and then the development of chemotherapy. Now, surgery and radiation are localized treatments. They treat the tumor you see. They don't really treat the tumor you don't see to a great extent. Immunotherapy and chemotherapy are able to potentially treat metastatic diseases. And now we have the new generation of targeted chemotherapies that I won't be talking about, I just pointed out. Okay, so here's a few concepts in tumor immunotherapy that are worth understanding. The first is that 20 years ago, most immunologists thought that the tumor was not recognized by the immune system. That's clearly incorrect. What's happening is that we develop microscopic tumors. Many of them are eliminated by the immune system, but the ones that are not eliminated by the immune system are actually able to suppress the anti-tumor immune response. And in this way, they survive and they can metastasize. So I think that it's become increasingly clear in almost every medical discipline that the immune system plays an important role. The conference is to discuss ideas of multidisciplinary treatments, multidisciplinary ideas, and I think in almost every one of those you're going to find a role for the immune system. So this is the role for the immune system. Basically, it's being suppressed by the tumor. So then the question would be, if you can stimulate the immune system to break that immunosuppression, can you help treat the disease? So a lot of people are trying to do this by doing systemic immunotherapy. They put in various compounds, drugs, cells, manipulated cells, systemically. We're taking a different approach. We're saying, okay, the immune system is inherently able to systematize a response. So if you can develop approaches that stimulate a systemic immune response by treating the tumor you see, whether you know it's metastatic or not, if you can treat it in an immunostimulatory manner, and then potentially follow by surgery rather than immediately taking it out, we might get better outcomes. So that's the overall idea, and I'll show you two of the experimental ways that we've been working with it. The first is using local hyperthermia, heating the tumor. This causes damage to the tumor, changes a variety of the cellular and biological parameters of the tumor, and helps break the immunosuppression. And the way I'll be showing you that we're doing it is with iron oxide nanoparticles and an alternating magnetic field. We also are going to show you a little bit of data on another idea. It's actually a very old idea, and it goes back to William Coley. There's nothing more immunostimulatory than microorganisms. We understand that. We understand. fire fast and they heat up. They absorb the energy of the magnetic field and they heat up. So it's basically one of many ways to control it. We'll look at two different models. One that we would call an immunogenic tumor, meaning that the immune system can relatively easily recognize it. And another that's very poorly immunogenic, D16, and it's very difficult to see for the immune system to see. So what are the immunostimulatory properties of heating? One is you're destroying some cells, you're generating exosomes, you're generating heat shock proteins. Those are being influenced dendritic cells. Dendritic cells present antigens to T cells. So you're really manipulating the antigen source, the antigen availability, the antigen presentation. They also become more visible to cells that have receptors for NK ligands, like NK cells. The heat will increase the expression of the ligands, or it can also increase class 1, which is recognized by CD8s. The last possibility is that you get increased vascular permeability. So here's some data. Put a tumor on each side of a mass. Inject particles into one and heat that tumor. This is a control animal. The tumor that's heated stops and disappears. But what's important to us is this is what happens to the tumor that's not heated. This is the tumor on the other side of the mass. So this is a control animal that had no tumors heated. This is the response of the unheated tumor on a mouse that had a tumor that was heated on the other side of the animal. So this tumor did not get heated. We know that. The immune system is responding. Okay, we're going to switch the model to B16 because it's a preferred model for immunologists because it's difficult to treat. And we're going to switch the pattern slightly. So we're going to establish the tumor. After the tumor is established, we're going to put in the particles and heat it. We're going to wait 3 days and then surgically remove the tumor. And then we're going to re-challenge the mouse on both sides. The control animal against a re-challenge. It's a good model for metastatic treatment. It turns out that in this model, the amount of heat is very sensitive. If we use a bit more heat, and I won't go into the physics of how the heat is really quantitated in a biological manner, but basically, rather than 43 degrees for 30 minutes, we heat for 45 degrees for 20 minutes. And it turns out, if you understand the science of hyperthermia, that's about a 4-fold higher dose. So it's a significantly higher dose. But what's important is the re-challenge has no protection. The primary tumor will be completely destroyed. That's very nice. But we're not getting any immune response against the secondary tumor. So what it tells us is we have to have a certain amount of viability left in that primary heated tumor. It's not been completely destroyed. We look at the cytokines. So we're immunologists, we look at the cytokines. We use a large panel of what's called a Luminex, and we see lots of expression of cytokines, and it's very non-specific. Virtually everybody goes up in the tumor after the heat. So it's got a strong effect, but it's not very specific. We look at the types of cells that are required. If we use an antibody to remove the CD8 T cells, starting from before the heat, we lose the effect. It goes up into this line. So we know we need CD8 T cells. A few more things we know about it that I'm not going to show you the data is if we use an antibody to remove cells that have NK 1.1, we remove NKTs, we don't need them. We don't need IL-12, and that's a bit surprising for you who are pretty knowledgeable about tumor immunology. We don't need CD4 T cells, so it's a relatively narrow result as to what we need. This is the basic model, the idea. work on the primary tumor or find potential metastatic disease. Okay, into the tumor. As I said, it was really pioneered by an American surgeon in the early 1900s. Microorganisms are the ultimate immune stimulators. There is some current usage for it in the U.S. BCG, which is attenuated mycobacteria, is instilled into the bladder after surgery for superficial bladder cancer to stimulate an immune response. There's a variety of research being done all over the world using genetically manipulated viruses, bacteria, and parasites. I'm going to show you a bit of data in which we're using a parasite, Toxoplasma Gondii. It's an obligate intracellular parasite. It's also the most common mammalian parasite on the planet. We call it the CPS strain, and it's got a mutation so that although it can go in and invade cells, it can't replicate, so it can't actually infect. The patient can't be infected, so it's completely attenuated. And as you see on the slides, we call it CPS. Okay, so this is the type of experiment. We grow a tumor, inject CPS on a couple of days, let it rest, do it again, and track what happens. Untreated, this is mouse melanoma. You get a large tumor by day 21. Treated, the tumors disappear. Here's the growth curves. Occasionally, a treated tumor will reappear. This is the survival curve. Okay, that's nice, but we're really not that interested yet in treating the primary tumor. But what do we know about it? One thing we know is that it's not simply stimulation of pathogen-associated molecular patterns. It's not simply toll-like receptor stimulation. We need live microorganisms. If we heat kill the microorganism, we lose the effect. If we have a mouse with no immune system, we lose the effect. Right? So even on that primary tumor that's got microorganisms pumped into it, it's an immune response that's taking away that tumor. Okay, this is the punchline slide. It's a little bit complex, but this is really what it's about. How do we get systemic immunity with this? So if we start the treatment, and then we do a re-challenge on either flank, finish the treatment, controls are once again removed the day after the treatments are finished, so those are our controls. When we re-challenge, we get growth unless we've treated. So these are the control animals. 100% of the re-challenged mice that have had the treatment and are re-challenged in the middle of the treatment reject. 100%. We've done it many times. We've never had a mouse that got the tumor. Let's look further out. This would be day 40, which is during the effector phase. The T-cell effector should still be around. 2/3 of the mice are able to reject the re-challenge. The other ones grow more slowly. How about immune memory? We go further out yet, 120 days. This would be immune memory. None of them are able to reject, but they all grow more slowly. So we have some memory, but it seems like the key thing is we have the strongest impact when the treatment of the primary tumor is being done. And that's metastases are already going to be there. They're already there. So if you have a peak of early in the treatment, that's fine. Okay, then we took one of these mice that a dermal melanoma re-challenge. And this was a one-time experiment. We had 3 mice. We did it with, and we waited for 2 weeks, and we re-challenged them IV. This is a model that will go to the lungs, form metastatic model-like tumors. Not true metastases, but they'll grow in the lungs. And what we found is after about day 30, this is what the lungs of the surgically treated animals look like. They're dying. We took our first of 3 mice on day 32, no tumors. We took the next one on day 65, no tumors. We took the final one on day 150, they never got tumors. So the metastatic re-challenge was perfect. We haven't followed up. It's a recent experiment, so we'll see how well that goes. And I think I'll just thank my various collaborators. A lot of the work was done by 2 graduate students, Seiko Touré Brown and Jay Baird.